Good morning, and I welcome you back to the channel. As usual, it's beautiful having you here. I will just discuss something very quickly today. We're going to look at identification techniques for bioweapons. We've been doing this discourse on forensic microbiology, and we've been talking about bioweapons for a while. So we're going to ask ourselves if there is the outbreak of, if you have a biocrime or the outbreak of bioterrorism, and you as a scientist, you're called in, along with government agencies, the police, the secret police, and all of that. And you, as a scientist, you're looking what are the techniques that you're going to use, to identify a bioweapon. What are some of the techniques that we use in microbiology to be able to identify bioweapons? The first technique is microscopy. As a microbiologist, as a scientist, you must be able to make use of your microscope. You must be able to work with it and know what exactly you're doing with it. So the first technique is microscopy. And in microscopy, we're going to make use of the microscope to try to identify the type of organism that is being used as a bioagent, as a bioweapon for bioterrorism. So we're going to have stains. In microbiology, we have different kinds of stains, GEMSA stains, acid-fast stains. That's a whole story for another day. So there are different kinds of stains, and then we will prepare our organism, fix it through many methods. And when you say you're fixing it, you're just making it stick more to your slide, and then you prepare the organism, and then you stain it. When you stain it, you mount it on the microscope, and then use different resolutions of your microscope to try to. And what are you trying to do? You're trying to look at the image. There's a word we use, morphology. So when you're talking about morphology, you're talking about the shape, the size, what does it look like? Is it circular? Is it oval? Is it cocoid? Is it like bar-shaped? Is it like a zigzag shape? So these are investigations that we do under microscopy. So you want to use your microscope to find out what is the shape, what is the size of this organism of interest. So that's microscopy. That's the first technique that we use in trying to identify what kind of agent is this bioagent. Now, the next thing that we are going to do is, or maybe I should have said one before the other. First, we are going to culture. So you want to culture the agent. Now, if a bioagent is used for terrorism, you want to culture it. And the issue with culturing is that it could also present a lot of difficulties because there are many issues. Number one, not every organism is culturable. We have a lot of microorganisms that are not culturable, and they are not culturable for quite a number of reasons. One, we might not in the laboratory be able to simulate the same conditions that the organism will have in the environment. So you may not be able to culture. Then we have what we call culture media. It could be in solid form, agar, it could be in liquid form, a broth. And so we use this media, this culture media, to culture these organisms. In plain language, we are growing them, making different cells, many cells of the organism. And why is it important to be able to culture microorganisms if you can? It's important because, one, you have a microbial population that you can study. It's difficult to study just one organism, but if you have them in tens, in thousands, in hundreds, then you're able to study the whole population. You're able to look at the size. If you have so many in a cluster, then you can look at their shape. You can look at their size. You can look at how they react to different reagents. So that is the essence of culturing microorganisms. So, culture is an old technique, but we still continue to use it today because it still, presents us with a lot of information. So you culture the organism if possible. There are different kinds of culture media that can be used to culture organisms, your EMB, your MRS, your Salmonella Shigella, your nutrient agar. There are so many. It's a message for another day. So many media. So we culture and then try to study, try to subject the microorganisms to different types of conditions. And the idea behind that is to try to identify. Remember that what we're trying to do is to try to identify the microorganism. What kind of microorganism is this microorganism? So first, you can culture. After culturing, you can mount on the microscope, stain, and then, you try and view with the microscope. The next kind of technique we use is PCR, polymerase chain reaction. Now, this is very popular, and this is going, we're proceeding to the molecular level. And when we say polymerase chain reaction, what are we actually doing here? What we are doing is that, think of it like tagging. When we talk at the molecular level, we are talking about sequences. Now, at the molecular level, the DNA exists. The DNA is in a double helical structure. So you have two strands intertwined with one another. Now, those strands can be separated, number one, so that you have individual strands. Now, apart from separating the strands, number one, you know that on each of those strands, you have nucleotides: adenine, thymine, cytosine, guanine. These are nucleotides, and they stretch along those strands from up to down. Now, you have very long stretches of strands and very long sequences. Now, to be able to identify an organism, you have I don't want to go too deeply so that I don't digress from forensic microbiology, but it's important to know that you have these sequences. And not only do you have these sequences, you have what we call a 16S rRNA gene, which is a unique gene that can be used to identify organisms. It's unique for different organisms. That means the sequence, the setup for different organisms is unique. What do we mean by is unique? It means that for E. coli as an organism, it has its own unique set of sequences. So you can identify E. coli, you can identify Acinetobacter baumannii, you can identify Klebsiella, you can identify Streptococcus, Staphylococcus. All these organisms you can identify because they have their unique sequences. Now, what is PCR doing? PCR is trying to look through an organism and trying to tag. So you have what we call primers. And I'm going so far now. You have what we call primers, and primers, you can look at them as your tags. So you have forward primer, you have reverse primer. Now you can use those primers to tag the sequence, tag it at the beginning and at the end so that you can use it to identify it. So the idea is this: let's say you have 20 sequences and you're trying to identify a particular organism. Then you have the primers for identifying that particular organism. Now, if you have 20 of those different sequences, you run them. And once the tag is able to match at a particular point, both forward and reverse, then you can say that, okay, based on the primer that I have, which I know that this primer is for E. coli, or this primer is for staph, or this primer is even a universal primer for universal identification, whatever the primer is, you can now use it and say, okay, based on this my primer, if it tags any of the sequences that I'm dealing with, then I know that that sequence is for identifying that organism for which I have the primer. I hope I've been able to give you a little bit of understanding of what the PCR. So the PCR, in essence, what it does is it makes many copies of the DNA, amplifies the DNA, then we use it and use primers and tag at those sequences to be able to identify which one is for what organism. So this is a simple technique that we use. So we use microscopy, we use culturing, we also use PCR, which is important for us to know. We also use what we call ELISA. When you talk about ELISA, you're talking about serological tests. When you're talking about serological tests, you're talking about antibody-antigen responses, reactions. And these are important because we use it a lot in microbiology and biochemistry. So you have ELISA, which we refer to as enzyme-linked immunosorbent assays, which are different kinds of tests that can be used. Some of them are even rapid tests, that's RDTs. They can be used to identify organisms. So you already have a unique identifier. You have an antigen, which you have bought, in the form of an antisera, or depends. There are many tests, but let's go too deep into it. Realize that you have ELISA, and ELISA is also used as a technique to be able to identify organisms. You also have what we call mass spectrometry. So we have different kinds of equipment, but we have an equipment we call the MALDI-TOF, and the MALDI-TOF can also be used to identify microorganisms. And how does it identify microorganisms? It uses what we call a mass to charge ratio of the protein of a microorganism. What do we mean? What we mean is that every organism has unique proteins that are unique to that organism that can be used to identify that organism. When I teach students, I say things like, if, for example, in your family, you have a particular shape of head, that shape of head can be used to identify members of your family from generation to generation. It means that 30, 40 generations from today, if they see that shape of head, they use it to identify your family. So that is sort of used also in science. So in this instance now, we're talking about a unique identifier protein. So there are proteins that are unique to microorganisms. And using laser, it is possible to ionize proteins that are present in a microorganism. That means you change the proteins to ions, positive and negative ions, and then use those ions to identify the organism, because you have databases where you match the proteins that are present in your bacteria of interest with proteins that have been sequenced and put into that database. So this is a method that is used, that is the use of mass spectrometry. We use that technique also in identifying organisms. And lastly, there is sequencing. We have high throughput sequencing, we have Sanger sequencing, we have 16S rRNA sequencing. These are all different molecular methods that are in play today to use to identify microorganisms. So I've run you through the techniques that we use in identifying microorganisms in when we are doing bioterrorism investigation. So in essence, what we are saying is that if a biocrime has occurred, if bioterrorism is in play in a particular country and scientists are called in, what are the techniques that these scientists are going to use to try to identify what kind of biological agent or its toxin or its spores are currently being used for this crime or terrorism? So they could try to culture the organism, they could mount it on a microscope, try and stain it and try and view what kind of microscope is. They could run PCR to try to identify what kind of organism that they are dealing with. They could do different types of sequencing to try to identify the organism. They could run ELISA to try to identify the organism. They could do MALDI-TOF to try to identify the organism. So these are different identification techniques that are in play and being used by microbiologists. Thank you for listening this far. I hope you share this content and help our students to learn. Science is fun. It's interesting. You just need to put your mind to it. There's nothing difficult in it. It's interesting and it's fun. Thank you for being with us today. You have a beautiful day.